Welcome back. Uh, in the last session, we have given a brief introduction of the drug discovery processes, and we have seen that there are several stages in a drug discovery process. Now, even a drug can fail, there is a high failure rate, and high failure rate, and that is because whatever you see in the test tube, that is not reflected in the in vivo studies, and whatever you see in the preclinical studies may not be reflected in the human subjects. So, that is where the drugs can fail. Okay. Now, when a drug, a molecule, an intended drug molecule is taken up to the suppose it crosses the initial stages, as I said, drug this target identification, target validation, followed by drug in silico studies, and then from in silico you get some molecules, and then you synthesize that, and then you see the actual interaction by a bioassay technique, and then you try to find out what are called hits, that means some preliminary effect you need, and then you tinker around the structure, and finally, you get some lead, and the lead has to be optimized through what is called I did not mention that structure activity relationship, that you make a large library of compounds, and then you try to find out a structure activity relationship. That means, suppose there is a phenyl group at some point, and then you replace the phenyl group with electron donating aromatic ring or electron withdrawing aromatic ring, and you see what is happening with the effect. And if you sometimes the electron donor may be helpful, sometimes maybe electron withdrawing may be helpful, but that is what will give you the structure activity relationship. Okay. That means, the activity how does it depend on the structure at different segments in the molecule. So, that will help you to identify a lead, and then the lead has to be now taken into the preclinical trials, and preclinical trials is basically done on animals, and in animals you have so you study the toxicology. Some people say that better first study the toxicological effect, because even if you have good ADME properties, you have good PD properties, pharmacodynamics, but then once you have already invested money on this, and then you see the toxicological effect, and you find that it is very toxic, then the whole money that you have put is gone. Okay. So, better, some people say, or some medicinal chemists say that better study the toxicological property first, and then you proceed to study invest money on other type of effects. Now, toxicological effect is basically, as I said, all drugs are ultimately toxic, but that they are toxic at a certain concentration. At lower concentration, as it is a drug, we can assume that it is having some good activity, but as you slowly increase the concentration, you might see that toxic effect developing, and in many occasions it may also cross the lethal. That means, it is a lethal compound, that means, it can kill the subject. Okay. Now, here in this graph I have three sigmoidal curves. Okay. The first is basically at you are giving a certain lower concentration of a drug, and you are measuring the response. Response means, you have, suppose if it is in the animal stage. So, you have different animals, 50 animals or 100 animals, and you are giving this drug to these animals at different concentration. Okay. And so, you will see the effect, this is the average effect on the animals. Okay. Now, so basically at lower concentration there will be very less effect. So, it actually picks up in this region, and the middle point of this what is called the ED50. ED50 is what is called effective dose. Effective dose, that means where you have, you do not take this dose as the effective dose. The effective dose is somewhere in between this point and that point. That means, here it is the 50%. That means, if you have say 100 animals and you are slowly increasing the concentration of the drug, if you see that the desired effect you are seeing in 50% of the, in 50 animals, then that means the concentration needed to have the effect shown by 50% of the subjects. Okay. That is what is called effective dose. If it is animal, then if it is animal, effective dose in animals. If it is in the clinical level, then it is the effective dose on human. Okay. So, there is a term called effective dose, or this is also called the therapeutic dose of a drug. Now, as you increase the concentration, you start seeing the toxicity. Okay. The toxicity will be definitely will be shown at a higher concentration, but it also follows a sigmoidal curve. And there is also another terminology or another parameter which is called TD, that is called the toxic dose. Okay. The TD is called toxic dose, ED is called the effective dose, and LD is a lethal dose. That means, if you cross the toxic dose, then you have a lethal dose. So, people so the subject will be killed. Now, we have ED50. As I told you, this effective dose, that means the dose which gives you the actual the effect that you wanted, is between here and there, and here it is the 50% of the population is showing the effective dose. Okay. Or showing effective response. So, there is this, we have, according to that, we have ED50, we have TD50 and we have LD50. ED50 is the dose at which 50% of the population therapeutically responds, because we know that the same aspirin, I give you an example, same aspirin causes acidity to a person, but may not cause acidity to another person, may have very good analgesic effect. Okay. So, if I have 100 persons and then I give this aspirin to several of these people, and then if 50% of the people are saying that yes, I am having this good effect, that analgesic effect. So, that dose what is called the effective dose. Similarly, you have TD50. TD50 is the dose at which 50% of the population experiences toxicity. Okay. And then you have lethal dose, dose at which 50% of the population dies. Okay. That means, it is not that at a particular concentration, like this point is what is the LD50. If you cross this, then all the subjects will die. If you cross this point, that means this concentration, but in between these two concentrations, in the middle of that sigmoidal curve, that gives where 50% of the population dies. Okay. So, these three things are measured. Usually, animals are sacrificed. So, you can go to the LD50, that means, you can see what is the dose required in which the 50% of the animals die. Okay. So, for animals you can measure ED50, ED50 you can measure, and you can measure LD50. Now, this ratio is very important, this LD50 by ED50. If the ratio is very high, that means you have a very safe margin, you have a safe margin. That means, suppose we are talking about this type of graph. So, suppose this is your effective, this is the effective dose curve, and if your lethal dose curve is also very similar. So, there is not much therapeutic safety level that you have. If this goes here, then you have a better window, that means, there is a less chance of crossing the lethal dose in that. So, this ratio, that means LD 50 by ED 50 is what is the therapeutic index. Initially I told, when I began this, I told you about this. So, therapeutic index is, there are two ways of doing this. For human, you cannot kill the human. So, for human you just measure the toxic effect, that when you slowly increase the dose level. So, some point you will see toxic level appearing. Okay. So, you see that what is the dose required to have 50 the toxic level in 50% of the population that you are that you have taken and you are studying. So, TD 50 by ED 50 is called therapeutic index. So, also LD 50 by ED 50. LD 50, ED 50 is usually done for animals, because where you can sacrifice the animals, but for human it is usually the TD 50 by ED 50. But anyway, this is what is called the therapeutic index, and the larger this therapeutic index, the safer is the drug. That is most important. So, this is an example. Say this is the, suppose this is the scale, and this is the amount at which drug is effective, and this is the amount the drug becomes toxic. That means, you have a safety margin of this. This is what is basically the therapeutic index, but it is expressed as a ratio of the TD 50, that means the toxic dose TD 50 divided by ED 50, or LD 50 divided by ED 50. Today also there is another, the margin of safety. You can even be very sure that that a person does not cross or is not given a dose which is a toxic dose. So, to do that, there is something called margin of safety, MOS, which is calculated by TD 1. That means, 1% of the population is having a toxic dose, and ED 99, that means 99% of the population is having the effective, showing that response, effective response. So, that gives you a better, even a safer margin, because you do not want only even 1% of the population to be affected by the toxic dose. So, rather than going up to 50% of the population, it is basically you increase the effective dose for the 99% of the population, and you reduce the toxic dose for only 1% of the population. So, that gives what is called the margin of safety. As I think these are all done. Clinical trials, I told you, clinical trials are basically, there are phase 1, phase 1 is safety, tolerability. There are different targets for this phase studies. Rather than efficacy, the first, as I said, safety they try, tolerability, that means what is the tolerance limit, and then the bioavailability, that means the ADME properties, that whether the drug is absorbed and then shows effect. Usually, healthy volunteers are used for the trial period, because you are only checking the safety level, you are only checking the tolerability, and you are only checking the bioavailability. You are not focusing on the disease yet. So, first this is what is done, healthy volunteers are required. Now, this is many hospitals this is done. I know in Johns Hopkins University, I know people are paid for acting as healthy volunteers to participate as trial participants. Okay. So, but this is safe, because they give very minimum amount of the molecule of the drug, intended drug. Okay. So, once these are through, that it has got a very good safety margin, those things, then you go to phase 2 clinical trials. Phase 2 clinical trials, you actually take the patients now, several hundred, suffering from the condition that the drug is intended to target, and then you try to, you get information about the efficacy, and also you estimate the safety in a larger population rather than the healthy subjects earlier in the phase 1 clinical trial. So, basically phase 1 is on healthy volunteers in a, so it measures the safety, that means it actually concentrates on determining the toxicological effects and the ADME effects, that means the absorption, desorption, metabolism and excretion, those type of, but no PD, that means we are not targeting the efficacy of the molecule. Efficacy of the molecule is done on patients, but in a limited way, not thousands of patients, in a limited way, and then try to find out the minimum amount required to get the efficacy, the dose that is required to cure the disease. Okay. And then phase 3 is basically phase 3 is, if assuming satisfactory results from phase 2, the drug will enter phase 3 clinical trials. These are larger versions of the previous trials. That means, now it is a broad based approach, but all are the patients as well as patients, you take the patients, you give the drugs which cross the phase 2 trials, and then you actually go to the patients, large number of patients, and give these drugs, and then see the risk and the benefit analysis. Risk and benefit, that means the toxic effect versus the benefit. As I said, every drug has a toxic effect. So, but the beneficial effect is very, you have to see whether ultimately this cost, this risk and benefit, which is better, which is more important to you. Like some people are taking anti-hypertensive drugs. Okay. Now, many of these anti-hypertensive drugs in the long run is going to affect the kidney, but whether you want to live 20 years from now on, take a drug which will reduce the blood pressure, or a person can die any time if he does not take the drug. So, this benefit and risk you have to calculate, okay? And then finally, the regulatory authority approves the drug. There is something which is called phase 4 clinical trials, which is that after this trial, phase 3 and the drug is available in the market, still many pharmaceutical companies then also study a wider variety of people, variety of patients, and see that in the next 5 years, 10 years, is there any, what is the report, what is the report of the patient's report, how are they feeling, whether they are reporting something else which was overlooked at that time. Okay. That is what is called phase 4 clinical trials. In many cases, some drugs have been approved, it entered the market, and later it has to be withdrawn because they are having some other effects which may be detrimental. Okay. So, this is what is the drug development process: validation, assay development, high throughput screening, hit to lead, and then lead optimization, preclinical, and then clinical drug development. There are some empirical rules when you design certain molecules for in silico screening. It is not that you write randomly whatever you would like to do. You have to follow certain things, like time and again I am saying that drugs are low molecular weight compounds, but what is the optimum molecular weight that you should not cross, or what is the threshold, like lethal dose which you should not cross to have a lethal dose. Similarly, molecular weight, there is some restriction, on the lipophilicity, that means how soluble it is in a lipophilic solvent, okay, like organic solvents. And then for interaction with the binding site, these interactions are electrostatics or could be hydrogen bonding. Okay. Hydrogen bonding plays a very dominant role in these interactions. So, how many hydrogen bonds you need in the molecule to be there. Okay. So, Lipinski made some rule which are called rules of Lipinski's rule of 5, Rule of 5 does not mean that there are 5 rules. Rule of 5 means that whatever numbers are shown here, they are multiples, they are either 5 or multiples of 5. Okay. So, Lipinski's rule of 5 says that whenever you select a molecule, you are thinking that it may be a possible drug, first check whatever you have written is molecular weight is greater than 500, then discard that. It should be preferably, it is not a sacrosanct that it has to be less than 500 always. There may be some molecules which are having 550 or 600, but this is a general guideline that a molecule is drug like when it has a molecular weight of less than 500. It has a log p value of less than 5. What is log p value? It is the partition coefficient, logarithm of the partition coefficient between octanol and water. Why octanol? Because octanol mimics the membrane through which the drugs crosses. Membrane is actually lipophilic. This is large chains, this fatty acyl glycerol chains and with a polar head group, but a majority portion in the membrane is lipophilic. So, what happens? The drug has to cross this lipophilic membrane and then go inside. So, that means it should have some balanced lipophilicity or hydrophobicity. Okay. So, here this is log p is the lipophilicity, that means the logarithm of partition coefficient between octanol and water, and octanol it was found that it resembled the biological membranes. Okay. And then it should have maximum, means less than or equal to, I can say less than or equal to 5 hydrogen bond donors. That means the sum of hydrogen bond donors means that usually OH and NH. So, sum of NH and OH in the molecule should not cross 5. Okay. It could be 4, but the threshold value is 5, and then the 10 hydrogen bond acceptors. That means, what are hydrogen bond acceptors? Nitrogen lone pair, free nitrogen, not NH, and oxygen lone pair. So, oxygen is a hydrogen bond acceptor, OH is a donor, NH is a donor, and N, a tertiary nitrogen, is an acceptor. Okay. So, this is what is called Lipinski's rule. So, now all pharmaceutical companies initially screen the molecule based on the Lipinski's rule and reject which does not satisfy this type of parameters. Okay. Remember, this is only true for oral drugs only, because we are talking about log p value, we are talking about this molecular weight, we are talking about this hydrogen bond, all these things. This is for oral drugs only, and a majority of the drugs we have are orally taken. Okay. Only and the life saving drugs are usually given, many of those are intravenously given because of their poor bioavailability and absorption. Okay. Now, let us go into some of these. We have crossed this. Ajab, before that, I think I tell you about some of the references. There is a medicinal chemistry book. The real name is Medicinal Chemistry and Introduction, which is by Gareth Thomas. Then Introduction to Medicinal Chemistry, just the Introduction to Medicinal Chemistry by G.L. Patrick. G.L. Patrick, that is a good book. And finally, R. Silverman's book on Organic Chemistry of Drug Design. Organic Chemistry of Drug Design. These are very good books on the topic that I will be covering. Okay. Now, I will tell you that already you now know that the drug discovery process ultimately in the initial stages, once the target is known and validated, then you have to make ultimately molecules, you have to synthesize molecules. That is where the organic chemists are required. So, they have to make the molecules, and one thing is very clear, the greater the number of molecules one makes, the greater, the more is the chance of striking a hit. Suppose you have 2 molecules and or you have made 5 molecules only, and these 5 molecules may not produce anything, because you have a very small pool of molecules. So, the larger the number of molecules, or larger than, this is called molecular library, the larger is the size of the library. That means, if you are looking for a book, and if the library has only 100 books, the chance that you are finding that book is very slim. So, if you have, say, if the library has 1,000,000 books, then the chances that the book you are talking about is there. Similarly, in drug discovery process, the more number of molecules that you make, the greater is the chance of success. There is no doubt about that. Okay. Because as I said, whatever you do, computational can assist you in making the library little bit smaller, but ultimately you have to make large number of molecules. Now, initially what happened, organic chemistry developed in a rapid stride, the synthetic organic chemistry in the 40s, 50s, 60s, Robert Robinson, starting from Robert Robinson, and then it went to then Woodward, and then many of the stalwarts in organic chemistry, they have now established the art of synthesizing molecules. So, organic molecules can be made, but how many molecules you can make per day? Usually what? You do 1 reaction or 2 reactions per day. So, one person 1 or 2 molecules you can make per day. What happened after the 1970 or 80s, maybe after 1980s, there has been an enormous development in biology. And earlier what happened, the biologists were, suppose some biologist has isolated an enzyme, and the amount of enzyme he or she has is very little, and then he or she wants to check whether what is the interaction of different molecules with this enzyme, new enzyme. Now, the chemist will supply one compound, and the assay system was such that it takes about few days to ultimately come up to a conclusion whether the molecule has really interacted, how much effective was the interaction. So, it is taking long time. But after the 1980s, something came which is called high throughput screening. That means, you can screen several hundred molecules with a very short time. In a very short time you can screen, and that means now gone are those days where the organic chemist synthesize one molecule and give it to the biologist, and 1 month later get the result that nothing has happened. So, you make another molecule. So, things are very slow, but then after this high throughput screening, that every day you can screen large number of molecules. So, that immediately put pressure on the organic chemist to come up with methods which can produce large molecules per day. So, that can be supplied to the biologists. Okay. Now, this has given rise to what is called combinatorial chemistry. Combinatorial chemistry was developed to produce large number of compounds required for high throughput screening. It allows the simultaneous synthesis of a large number of possible compounds that could be formed from a number of building blocks. The products of such process are known as combinatorial library. Libraries may be collection of individual compounds or mixture of compounds. This is very interesting. Libraries may be collection of individual compounds. That means, suppose you have 10 test tubes. Okay. You have 10 test tubes, and each test tube 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Now, suppose I add here say CH3 COCl. I add here I take CH3 COCl in all these test tubes, suppose the same acid chloride, and then I add different amines. Suppose this is methylamine I add, and here I add ethylamine, CH3, CH2, NH2, then propylamine and all this. So, in each test tube at the same time, because I am doing in parallel, so I will get the amide. In this case, it will be amide of methylamine and acetyl chloride. In this case, ethylamide, in this case, propylamide or propionalamide. So, you will get all these different amides. So, if you do the reaction in this fashion. So, here basically each test tube has only collection of individual compounds. That means, each test tube contains only one pure compound, and the other is mixture of compounds. That means, basically what you are doing, you are adding both methylamine and ethylamine, something like that, into a test tube, particular test tube. Okay. So, you get two compounds, one is the amide of the methyl, another is the amide of the ethyl. Okay. Now, what you do, then you do the testing. Now, chances of getting more success if you have more number of molecules in a particular test tube. Earlier case I said that 10 molecules in 10 test tubes, but there are techniques by which you can make 10 molecules in a particular test tube. So, that you have 100, 10 into 10, that means 100 molecules in these 10 test tubes, and then you test through high throughput screening. But the question is, suppose this test tube shows activity, that means there are 10 molecules which out of which maybe one is showing activity. Now, you can go back and then individually synthesize those 10 molecules and then see which one is active, but this is much better option, because out of 100 there is more chance of getting a hit, and then you know which test tube is giving some activity, and you know what are the ingredients that you have added, and you try to separate them and then separately study those 10. So, it is much better option. Now, this combinatorial chemistry was developed by using solid phase synthesis, because ultimately it was realized that the solution phase chemistry that we have, solution phase, that is detrimental, because that will give you in solution phase you have to purify the molecules, you have to do chromatography to purify the molecules. Whereas, if the reaction is done on a solid phase, as we have seen in solid phase peptide synthesis, your purification is very easy, that you just take the solid beads and then wash them after each reaction, and that will give you, and then you detach it from the solid phase, that ultimately gives you the compounds. Okay. So, all this combinatorial chemistry are usually done on solid surfaces, okay, like this Merrifield, utilizing Merrifield's resin, like that. Okay. Now, I give you an example of this combinatorial synthesis. I suppose you take RCOCl. Now, different types of R which are exemplified by or denoted by A1, A2, A3. That means, what is A1? Suppose A1 is methyl, that means acetyl chloride, this is propionyl chloride, and this is say butyryl chloride. And then you are adding an amino acid NH2 with R dash, and then amino acid ester. Now, this ester, this R you can vary, you can take different amino acids. There are you can have 20 amino acids here depending on this R, and on the R dash, see these are your variables R dash and R double dash. So, you can vary this R dash and you can vary this R double dash also. And so, suppose I say that B1. So, basically if R1 is something, say methyl, that means alanine I have, and this is CO R dash, and this R dash is suppose the say methyl. So, that is exemplified by B1. So, similarly varying this R dash and R double dash, I can have another entity called B2, and then I can have a third entity called B3. So, what I do, I when I take A1 in one test tube, I add this B1, that means this is methyl and that is methyl. Okay. So, I will get A1 B1, and in another test tube I will get A1 B2 and then A1 B3, and regarding the A2 as the starting point, you will get A2 B1. You are adding the same components B1 B2 B3, A2 B2 A2 B3, and then here A3 you will get A3 B1 A3 B2 and A3 B3. I am not actually proceeding any further, but you see that the same reaction is operated on 3 different acid chlorides, and you get 1 2 3 4 5 6 7 8 9 compounds you get. Okay. I think solid support, this we can we know what is the advantage of the solid support. These are some of the resin beads. This is the, see, in Merrifield resin we know that it is polystyrene which is chloromethylated. Here the resin bead, then there is polyethylene glycol chain, that means you have this type of moiety. The beauty of this type of moiety is that they swell in water. Okay. So, if you have the resin bead, then polyethylene glycol chain and then a reactive X. Okay. As you put it in aqueous solution, this swells. Swelling is important because that allows percolation of the other entity that is reacting with it. Okay. X is NH2OH, SH, all this reactive functionality. There are other resins also. This is called Wang resin for carboxylic acid. This is the THP ether for alcohols, and then you have for amines you have this chloroformate type of resin. Means resins which are attached to a chloroformate, resin which are attached to a tetrahydropyran moiety. This is the tetrahydropyral linker, okay, and this is the Wang resin which is a benzylic alcohol. These are the resin that you will be used depending on your starting entities. Okay. Now, this is an example how this combinatorial chemistry is done. You have basically you take a plate and there are wells in the plate, that means grooves. Okay. Now, what you do? You are adding, suppose you are adding, you are doing this type of reaction, that this resin bead is attached to the via the ester linkage to an N-Boc amino acid, okay, with R1 and R2 as shown here. This is very similar to the peptide chemistry that we have done, and this free amine can react with an isocyanate, and that will give you a substituted urea. And it is known that if it is heated with HCl, then a reaction that takes place is this attacks the carbonyl and the resin is released. So, that makes say 1, 2, 3, 4, 5. So, a 5 membered ring, that is called a Hydantoin. Now, Hydantoins are privileged skeleton for drug development, because it has got this Lipinski's rule, this type of molecule satisfies. Okay. Remember, we have said the Lipinski's rule says the upper value. It is not that you have to maintain that upper value. You can have less number of hydrogen bond donors, less number of hydrogen bond acceptors, those type of things. Hydantoins are privileged skeletons. Okay. Now, what you do, I will show you in the possibly the next slide. See, these are my wells, all these grooves. Okay. So, I say that this is A, B, C, D, E, F, G, H on this row, and on the column I said 1, 2, 3, 4 up to 12. So, what I do? I add the first component. The first component is this bead with the NH-Boc. Okay. So, I add the bead with different amino acids with 8 different amino acids. So, I add X1, X2, X3, X4, X5, X6, X7, X8. So, these are the resin bead which are attached to this X1. Okay. And on this one I basically I added I took the bead which are having different attachments, different amino acid attachment. On this side what I do, I add the same bead with same I add the bead with same attachment, same amino acid attachment on this side. Okay. So, here it is different, the beads are attached to different amino acids, and in this direction, that means the column, I have the same, the bead attached to the same amino acid. Okay. Then this is, but these are different, as in the rows we have different ones, because these are x2s, this is x1 rows, x3, x4, like that. On this side it is x1, x2, x3, x4, x5. Okay. Now, what you do, you add your you deprotect the amino acid, the BOC, and then you add the next amino acid and couple. Okay. The next amino acids you have again, you have 8 different amino acids here, 8 different, not 8, actually these are sorry, these are 12, because there are 12 groups on this side, on the in the column. Okay. So, you have to select 12 amino acids and then couple with. So, Y1 now, earlier your x1, x2, so that means different, the beads are having different amino acids here, and on this side beads are having the same amino acid. Now, what you do, you add the second amino acid, the same amino acid on this side, on the row. So, you add the same amino acid on this row. So, what you will get x1 y1, x2 y1, x3 y1. So, y1 will be attached to all the beads via the x. Okay. And in this you add, so basically what you are doing in the second one, you are adding only y2. So, you get x1 y2, x2 y2 this. Remember, these are different. So, then you add the y3. So, in this case, if you add all these, so what you get is a library of compounds. How many compounds? Usually there are commercial plates available. These are usually 96 plates, usually 96 plates. So, 96 compounds you can prepare in one go. Okay. Another way you can do is, this is very very good technique, that you have a glass plate. Suppose you have a glass plate and there are, the glass plates are made of silica. So, you can actually, using the silica wedge groups, you can hook up an organic species ending up with a NH2. That is possible, that glass plates are available today, that where lot of NH2s are there attached to the glass surface. Means lot of NH2 means NH2 is not directly attached, it is attached through the that wedge and then an alkyl chain, and that ends up with a NH2. Okay. So, what this NH2 can do? This NH2 you can use for further reaction, and then that means your plate is the solid phase, it has got this NH2, then you are adding a compound which reacts with the NH2. So, the compound will now stick to the NH2. Okay. So, let us see what on this NH2, there is one scientist who did, a medicinal chemist, who developed this technique. What he did, that is glass plate with NH2, you react with a molecule which is called N-veratriloxy carbonyl. This group is what is called N-veratriloxy carbonyl, like your BOC, that is the butyloxy carbonyl. So, this group is what is called the veratryl group, the nitro sorry, this is nitro, I am sorry, this is nitro veratryloxy group. Okay. Nitro veratryloxy group, that is this. Okay. And then veratryloxy carbonyl, then you have the carbonyl. The beauty of this, that first of all it is a chloroformate. So, this is going to react. So, you form this what is called the urethane or the carbamate. Okay. And then as you shine UV, this is a protecting group which is labile under UV. So, it cleaves. Okay. So, it cleaves and the NH2 is again liberated. So, this is a group which is called nitroveratriloxycarbonyl, which also there is another group which is only a nitro and a benzyl, the similar group benzyl oxycarbonyl, orthonitrobenzyl oxycarbonyl, that also is cleavable under light, but this is a better one, because the wavelength of the light that is used for removal of nitroveratriloxy is around 365 nm. Okay. So, this is a photolabile group. So, you can again regenerate your NH2. Okay. Now, let us see how you can utilize this technique. So, you take a glass plate like this, and see, ultimate objective is to make library of compounds, and by a simple technique and then also a simple assay, and you can check the bioactivity. So, suppose this is the glass plate. So, what you do first, you cover one side of the glass plate with a opaque material, so that light cannot go through and fall on the glass surface. Okay. But the other two, you have made it actually 4 quadrants, and then in the 4 quadrants, 2 of these are covered by a opaque filter. Okay. It is not a filter, it is a opaque opaque screen, I can say, okay, an opaque screen which covers this part. Now, you shine, now what you do, this X is actually the NH2 attached to veratriloxy. This NH2 is basically, this is NH2 attached to veratrile, that N-VOC. Okay. So, all are attached to N-VOC, and this is also attached to N-VOC. So, now you have covered this part and shine light. So, what will happen? This will generate the NH2, because you are adding light. Okay. So, now you are adding light. So, this is going to form the free NH2, and you add the first element, whatever it is, amino acid suppose. So, you get G and G on this side. Okay. Via the NH2 you couple the amino acid. Now, what you do? You cover this part and shine light on this side, what was earlier covered by the screen, the opaque screen. Then what will happen? Then this will form the NH2 now, and you add the S, another second component. So, what will happen? Now, the plate will have GGSS. Now, what you do? You cover this part now, S and G part you cover, and you add the third, you shine the light, because your amino acid also had whatever amino acid you have attached to the amine, that is also protected as N-VOC. So, you shine the light, and so G will now G will have free NH2 after shining of UV, and so there is a G and there is a S. Now, you add the third component. Okay. Third component is here. You add the third component, suppose that is A. So, what will happen? You have a GA molecule, you have a SA molecule which are linked on the surface of the glass plate. So, the next step is you cover this part where SA and GA are produced, and then you add the AF, first UV and then add the AF. So, you have SF, GF, SA, GA. So, you keep on doing this, you mask this part, now you shine the light. So, F will have NH2, A will have NH2, you add the fourth component. So, in that way you can make these different compounds which are growing on the surface of the plate. So, if you have different plates, you can do it in series and then generate molecules on the glass plate, and on the glass plate also you can see the activity, that is what is high throughput screening. You can even, there are 4 compounds on the glass plate, 4 different compounds, and you can check the activity of these 4 entities okay, by high throughput screening. So, this is a very good demonstration of combinatorial chemistry. However, whatever we have done, that is what is called parallel synthesis, means we are after all, after these reactions, each quadrant here is basically homogeneous, that each quadrant will either have a GA or SA or GF or SF. Okay. It is not that every quadrant has mixture of compounds, that is not there. So, now there is another technique which is called mix and split technique, by which you make larger library of compounds. So, how it is done? You take the resin bead, okay, and you take 3 test tubes, suppose 3 test tubes. So, in these 3 test tubes you have this bead, resin bead. Now, you attach, you add A here, you add B here and you add C here. Okay. So, this will be attached to A, this will be attached to B, this will be attached to C. Now, what you do? You mix the contents of all the 3 test tubes. Okay. So, put all in a bigger test tube, everything is there. That means, some beads are attached to A, some beads are attached to B, some beads are attached to C. Okay. So, you mix and then you split it, you split again into 3. So, when you split into 3, so every test tube will have what? We will have a bead attached to A, attached to B, attached to C. So, you split into 3 test tubes and you add now D, and here it is E and here it is F. So, what will happen now? Here you will get AD, BD, CD, here you will get AE, BE, CE, and here you will get AF, BF, CF. Okay. Then again you mix and then split. So, what will happen? Each bead here will have AD, BD, CD, AE, BE, CE, because you are mixing and then splitting. So, every test tube have these characteristics of these earlier 3 test tubes. So, all these are there, that means there are 9, 1, 2, 3, 4, 5, 6, 7, 8, 9. Now, you add G here, you add H here, you add I here. So, what you have now? ADG, this all ends up with G, this all ends up with H and these all ends up with I. But how many compounds after 3 steps? After 3 steps you are having 27 compounds, okay. 27 compounds. So, you have a larger library. However, things are not that easy. So, what will happen now? These beads which are present in one test tube, you separate these beads, separate individually, because these are like marble. So, you separate those beads and then see whether the bead has given some activity or not. Okay. So, that means you separate it into individual beads and then check the activity. Suppose one of these, suppose this bead gives the activity and these beads give the activity, but the problem is you can isolate the bead. The problem is how to know what is actually attached here, because it could be ADG, it could be BDG, it could be BEG, CEG, all these things. So, that is the problem. So, you have to finally find out what is there which is attached to the bead. So, for that you have to detach it and then see what is there. I think in the next lecture we will start from this point. This is called the mixed and split method, mix and split method for combinatorial chemistry, and that gives a rapid growth, because if you further increase this, you know that it will be 81 ultimately, this will be 81 molecules you are handling. Okay. So, this is much better, only problem is later on you have to find out, you do not know basically, because every test tube has different collection, is heterogeneous. So, but you can isolate those beads and then see what is the effect and then finally try to find out what is the compound that is attached to here. Okay. So, we will discuss that in the next lecture. So, thank you for today.